Oh, this is I think our second annual or a second semi-annual research update for this year, and um, there's always so much progress. It's hard to decide what to discuss. So I thought, well, for some of the new families, I would talk about some of the amazing progress we have all made, our whole community has made over the last it's coming on 13 years. I'm going to have to update this slide for 2025, you see here. But it starts in 2012 when we discovered the gene that caused Shay's epilepsy, and she was the first. And then shortly thereafter, we started hearing about other kids generally who were diagnosed with epilepsy. And it started out as a fairly severe syndrome. And so I think that genetic testing was mostly um, approved for those with severe seizures that were undiagnosed. And the point I'm making here is that as has gone on, SCN8A has become more famous and more tested. So even for kids with milder forms of epilepsy, or even with kids with neurodevelopmental delays, autism, movement disorders, and things like that. So the spectrum has grown over the years as a result of the fact that it's being tested, approved for testing more broadly. And so what I'm going to try and tell you is that what's really helped us understand the spectrum is this international SCN8A registry that we started back in 2014, and it's been going now for over 10 years. And so, although we've attracted tremendous talent to the field, scientist-wise, clinician-wise, pharma has gotten involved and interested. There have been a tremendous number of publications that have taken off since 2012. I think a great chunk of research of progress has been contributed by the registry, and I wanted to give you some updates on the registry today and other projects that are going on that are associated with the registry and studies that caregivers are the primary input of data. So, because of caregivers giving us data and participating in research and staying engaged in research, we've been able to make tremendous progress. I really think it's really powered the field. And as you know, it's a tailored, there's several modules and it's tailored to the needs of almost everybody with SCN8A, some form of SCN8A. And we've had to add modules over time as we've learned about loss of function and different clinical challenges for loss of function. But the modules are very detailed. It takes several hours to fill out the first time, but then every time you come back and fill it up, it's an annual update. That doesn't take very long. It's really unique in that it's extensive, it's caregiver reported data, all with the goal of listed here, describing the full spectrum, the course of the disease, genotype, phenotype correlations, identifying patient subgroups. It's aiding in clinical standard of care. I'll briefly mention a landmark study led by Gabi and Jada and the International SCN8A Alliance, which was the first treatment and diagnosis and comorbidity study and consensus, and then integration with medical records. So we've been using the registry data to power all of this. You can see the caregivers at the top, which are the double arrows indicate it's a two-way street. We really try and give back to the caregivers that are providing all the data. And you can see that we have stakeholders in clinical areas, research areas, pharma, biotech. Many of them interested in the natural history of the disease. Well, going from Shay to now hundreds and hundreds of worldwide cases, I think there's over 1,000 that we probably know of right now in the literature and in clinics and through our own advocacy groups. And you can see that it's a very international, there's no boundaries when it comes to where SCN8A is found. Wherever genetic testing is, then we find individuals with SCN8A. There's about a 1 in 60,000 birth rate, uh, as estimated mostly from the epilepsy side. I'm sure it's higher now that we know about autism, loss of function, and intellectual disability. And those are things to be learned as we go forward. This is just giving you some basic demographics of our population. You know, it's the age of the mode of the age of our population is somewhere about 6 to 10 years old. But there are individuals that we know of that are over 40. Fewer of them because back in the day, people weren't tested. And I think once you've lived for a long time with a particular set of challenges, health challenges, you generally don't pursue genetic testing at older ages, but we do know of olders. There must be many, many, many out there that we don't know of. It's roughly male and female equally distributed, and 90% of people who have been diagnosed have seizures, but that number is coming down slightly as we learn about more loss of function. In fact, I think one of the things we learned from a research roadmap that I'll also talk about, we put out extensive surveys to caregivers. I think over 175 caregivers responded. And you can see seizures are an important concern, but many of the challenges that caregivers feel are cognitive, speech, motor, sleep, behavior, and many others. It's not just a seizure disorder. It's a very complex system that affects most of the systems in the body in some way. Another important finding, as I mentioned already, is that there are gain and loss of function variants, each with their own set of characteristics and features. On the left, you can see that 54% of people with loss of function have seizures, but the seizures are different in many ways from those with gain of function. Almost everyone with a gain of function has a seizure disorder of some type. So this is a major subdivision within the loss of function group, those with and without seizures. You can see some of the skills in this sort of word cloud that's supposed to look like an L for loss of function. And then over here, a G for gain of function. So they share many characteristics, but they also differ. And you can see there are many more comorbidities with the gain of function, and the loss of function are limited more to ADHD, autism, motor skills, hypotonia, coordination, delayed speech. But they all have delayed speech. They all have hypotonia. They all have some autistic features. So what we understood from the fairly early years was it was a spectrum disorder, shown by the arrow at the bottom, where you had no epilepsy, behavioral movement disorders with no seizures, moving into more benign forms of epilepsy that could be inherited, and to mild to moderate developmental and epileptic encephalopathy, which was a term that was not just coined, but popularized in the late 2000 teens. The International League Against Epilepsy took various forms of epileptic encephalopathy and combined it with the developmental encephalopathy to come up with DEE, and then the more severe form. So we knew there were a lot of different, a spectrum, but we didn't know whether patients fell into neatly distinct subgroups. And that's something that registries really allowed us to get a closer look at. The other thing the registries allowed us to do, and here's the topology. You can go on SCN8A.net and go to the topology second to see this and put your own variant to see where it is. But here's amino acid number 1. Here's amino acid 1980, 1980 amino acids in the chain that makes up NAV1.6, which is the protein that's encoded by SCN8A. SCN8A is the DNA, the gene. The NAV1.6 is the protein. The protein finds its way to the neuronal membrane. It inserts itself in the membrane. Now, this is still a two-dimensional view. You have to imagine it forms a three-dimensional pore to really understand what's going on. But this helps us see that there are certain variants in the gold stars that are found in more than one individual, which is highly informative for us to understand how much your variant contributes to your likely presentation of the disease. And I've just highlighted some very highly recurrent variants here. 850, Elliott has 850, 1617Q, 1475, 1872. There's a couple of different forms there, 1877. But there are other forms that are present in 5 or more individuals in the registry database. It's verging over a dozen now that we can really look at the features to see what's in common among people with the same genetic variant, which really helps us understand. This has helped us identify genotype-phenotype correlations. It's a busy slide. Sorry about that, but you can see here's, for example, the age at onset. You can see different variants tend to have different mean ages at onset, where the earlier onset variants like 850 are more severe, and the later onset variants like 1877 are more mild in terms of gain of function. And then you could see development follows developmental quotient actually shows that same pattern, the correlation between the age at onset and the developmental quotient associated with individuals with these variants. The number of medications shows the opposite. So if you have a later age at onset, a more severe variant, you have a milder variant, you're generally on fewer medications and you've weaned fewer medications. And then we also see really interesting correlations between individual variants and the what we call the electrophysiological score, which is determined by in vitro testing. So you might hear about this where people determine the biophysical properties of a particular variant in vitro. That means they construct that variant in a cell, or they look at it in a cell that's given by a skin biopsy or something by a patient in an iPSC. There are different ways to do this, but the electrophysiological scoring of that variant in vitro in a plate. It's not in an individual's body, but in a plate or a tube, and you can see that some groups have put together an algorithm to tell you how severe the variant might be electrophysiologically. And you can see it correlates very much so with an index that we came up with from the registry, where individuals, based on several different features, we could say if someone was more mild or more severe. And there's only one variant that falls off the line, it's 1872W, but everything else falls on a nice line. There's some interesting questions about why that falls off the line. And just to summarize, all the work that you have done in entering your data into the registry, the SCN8A.net registry on REDCap, has really powered a lot of publications in the last couple of years. You can see a whole bunch of publications here that I'm happy if you email me to send you a PDF if you don't have these. You can also go on to SCN8A.net and look for publications there as well. So it's not wasted time on your part or our part. It's really hitting the literature and making a difference. And this is the consensus I talked about in 2024. The SCN8A Alliance and its clinician network worked for 2 years. Much of this work, Gabi and Jayetta put their blood, sweat, and tears in to make this happen. We got clinicians from around the world. I believe it was more than 30. You can correct me if I'm wrong, Gabi, that worked together to find whatever consensus they could find on how to diagnose, treat, and prognose this disease. And at the time, I'm just highlighting one aspect of this. The registry made a big contribution to understanding the phenotypic subgroups, as well as other labs in the literature, like the Danish group played a big role. But you can see that we were able to subdivide patients into various groups, and the clinicians were asked if they could recognize these groups, but from severe, mild, moderate, self-limited epilepsy, very, very mild, that actually goes away, and then the loss of function. So the gain of function seemed to be more on this end, the loss of function more on this end, and there was very good consensus on these subgroups. And then the questions from there were like, what kinds of medications might work best for these subgroups? These are the things people are trying to infer. And basically for the gain of function subgroups, there was a very decent consensus for oxcarbazepine and carbamazepine as the preferred first-line treatments. It's just as far as they could get at this point in time. And so we really want to push this forward and reiterate this consensus process as we learn more, get better drugs, and have more information that will help clinicians decide how to treat patients. We've been, because our database is so large, we can no longer just look at the data and make inferences. We have to use computational approaches. So we use, some of you might have heard recently in Atlanta, Josh Hack gave an update on what he's been doing with the registry data using machine learning. And this just kind of shows sort of a general flow diagram of the history of what we've been doing. You input various features, including the genotype, developmental profile, seizure onset, seizure type. Our first machine learning algorithm was able to distinguish loss of function and gain of function without actually doing those experiments in vitro with electrophysiology, but to look at the properties of the patient themselves. So clinicians could eventually, or now using our algorithm, decide what the probability of a loss of function. That's a very important question for treating. You don't want to go with a sodium channel blocker generally if you have a loss of function patient. And many clinicians don't quite know when you have a genetic report diagnosing SCN8A, it doesn't always give the information about whether it's gain or loss of function. So sometimes they're still shooting in the dark when really that could make things a lot worse if they don't have that information. So we've worked on a way to do that computationally. Then the next step was to go from diagnosis to prognosis, and these subgroups emerged. We have a different way of looking at the subgroups than what was currently in the consensus, and I can explain that in a minute more. But there's two loss of function groups and at least three gain of function groups that we refer to in these simple terms: DEEE, DEE. And then the last step so far, and what Josh just reported in Atlanta, and what we just published this year in Epilepsia, a paper where you can decide which medications would be more beneficial for these different subgroups based on the data we had from you all in the registry. And it's a first step at trying to refine the medicine landscape, or the way doctors prescribe medications, to make it easier and more consistent. And I'll give you some updates on that. But the way we see it, so we were surprised when we looked at the phenotypic variation among patients. We had this idea that there were more severe, mild, and moderate phenotypes for the gain of function. But when we let the data tell us what it was seeing without us biasing the way we diagnose patients or put them into classes, it came up with something surprising. And that is, first of all, the landscape is not black and white. It's not like a checkerboard. There's blurred subgroup divisions. They're not hard lines. So we call it a partially ordered model, where an ordered model would be A is distinct from B is distinct from C. A partially ordered model is A could give rise to B, A could give rise to C, and there's a relationship between the three. And so we think that computationally and statistically, looking at this as a partially ordered landscape as opposed to an ordered landscape, which is more inferred by mild, moderate, severe, as ordered groups. This is a different way of looking at it. And basically, the bottom line is SCN8A is a developmental disorder with seizures added into the disorder. So every person with SCN8A, the common feature is a developmental impairment of some sort. Some patients are, even if they're loss of function or gain of function, they'll have seizures, and the seizures could be a problem, or they might not be a problem with respect to development. So there are groups that are developmentally impaired. We call that a developmental encephalopathy. They have seizures, but the seizures don't seem to be responsible for making the developmental impairment worse. So if you get rid of the seizures, which happens to be much easier in those with DE, the developmental challenges may remain to some significant degree. The other kind of encephalopathy we call an epileptic encephalopathy. Yes, there are developmental impairments, but they're mainly driven by how bad your seizure episodes are. So some patients do quite well developmentally, still impaired, still not neurotypical, but their seizures tend to be the thing that makes things worse. And if you get control of those seizures, you can get improvement in development. And the bad news is that sometimes those seizures are a little bit more difficult to control than in the case of the DEs. And then there's those individuals, unfortunately, almost half the population, something like 40%. We can't really distinguish the DE and the EE, and those are the true DEEs. They have a combination of developmental and epileptic encephalopathy. Both emerge very early, about the same time in life. Whereas in the case of DEs, you might find that someone's developing abnormally before the first seizure. And in the case of an EE, they might be developing just fine. They made all their milestones, and at 6 or 8 months, they got their first seizure. And lo and behold, there was some regression. So this is very important for understanding um, what our expectations should be and how best to treat some of these different groups. But prognosis is the main outcome from this kind of understanding. Now we want to know, are there different treatments that might apply to these different groups? And you can see the pie chart over here showing you the frequency of the different groups as it was at that time. Selfie being very rare. Those are the self-resolving epilepsies. The DEs and the EEs around, it's turning out they're both more around 15 to 20%, and then the DEs are more like 40%, and the two loss of function groups are a little bit more similar than shown here in terms of frequency. Okay, so now I just want to say we've made great progress. There's no doubt about it. This has been a tremendous run, but there's still a long way to go, as you all know. I'm just listing some of the things we don't know yet, and that's we don't understand the disease mechanism fully. I just published a paper, a review article in Bioscience Reports, where I put forward a model of epileptogenesis where it really focuses on the disease mechanism and it goes beyond the channel, like what happens in the cell, what happens in different neurons, what happens in the whole body as a result of SCN8A and other channelopathies, how they converge on similar phenotypes. Why would they do that? Well, there's an underlying set of mechanisms that are in common across all these epilepsies, and that's important for us to know. But we don't really fully understand it. It's just models at this point. We know of all these non-seizure phenotypes and comorbidities, but we don't really have a good way of treating 90% of them. Even a simple thing as sleep disorder, we don't know how to treat it, but there are many others, GI, pulmonary, cardiac, peripheral nervous system, autonomia, dysautonomia, many things. We don't know how to treat those, and we don't know what the common features are that may underlie them. Hypotonia is common. It may be part of a lot of these things, or dysautonomia, another common underlying feature, can explain a lot of things like heart rate and temperature and sensitivity, pain and sensitivity, all that, and even sleep disorder. We have a very high premature death rate. I think that within the years that we've been following this, over 70 children have died, which is roughly 10% of the population, 8 to 10% of the population. That's a very high. We don't know how to treat that. We don't know what to do about that. And really, a more esoteric seeming issue is biomarkers. Biomarkers are ways to track the progress of the disease without actually going to the doctor and saying, "Oh, I've had so many seizures," or "I've gained these developmental skills." Are there molecular or other physiological markers that tell us what's happening with respect to the progress of the disease that could be measured to get the better natural history, but also could be measured to say if we have new treatments, do we have to wait around for 2 years to find out if the treatment worked? Are there markers that we could look at incrementally along the way that we call biomarkers? So I'm very concerned about the low rate of seizure freedom. And, 85% intractability is a really high rate, even among other pediatric epilepsies, which normally have a intractability rate, or a rate in which medications just fail, two or more medications fail, of closer to 30%. Ours is 80%. So I want to focus a little bit more now on what we can do to improve current therapeutics. But I'm going to do that in the background of the research roadmap in just a minute. But I want to focus on why anti-seizure treatments are so ineffective. They help, but they're not perfect. And what are the best ways to treat this? And then to suggest at the end that one of the things we need, and we need your participation in, is to get better fine scale seizure frequency data in association with data on what medications you're taking and other life events that are happening that may tell us what the triggers are for seizures getting worse, but also what things are making them better, and what's the developmental consequence of making seizures better. Are we going to rescue many features of the disease by treating the seizures? That's the big elephant in the room. So along these lines, the International SCN8A Alliance organized the first research roadmap for SCN8A related disorders, and Madeline O'Don and Chris Burge were very big help in organizing this and situating it in Boston in their institutions, and played a big role in organizing it. And my heart and thoughts go to them right now because, as most of you know, Margot, their daughter, passed away just 2 or 3 days ago. And I'm sure it's a very difficult time for them. So prayers to them. And thanks to them also for all their hard work that they put into working on new therapies. And I'm sure that work won't stop. But back to what the meeting was in Boston in 2024, the summer of, uh, the purpose was to identify gaps in our knowledge with the goal of determining research priorities to fill those gaps. And then we had a great turnout of clinicians, researchers, families, industry, all came together for this 1.5, 2 day meeting in Boston. And we formed working groups to tackle these problems. And the 5 working groups that emerged were advancing gene therapies, both development of the therapy and how to deliver, understanding non-seizure outcomes, mechanisms, management, and therapies, improving current therapeutics. So while we wait for these advances in gene therapies, which we know are going to take years, what can we do in the meantime with respect to first line adjunctive combination and alternative approaches? Um, Again, how do we identify and apply biomarkers, as I described? And then another area is like, what's the relationship between SCN8A, the brain, and the whole body? We might consider the disease to be an epileptic body, not necessarily just an epileptic brain. So there's a ton of stuff that we don't know in understanding what cell types are involved, what circuitry is involved, what brain regions are involved. How do we treat those brain regions, perhaps with things that are not pharmaceuticals, but neuromodulation is another area of great interest that we are just dipping our toe into right now. So that was the overall look at the meeting. It did result in a publication. That was the goal, was to publish, and it did come out this year in the Orphanet Journal of Rare Diseases, and you can find it. Uh, and again, if you need a PDF, I can get it to you. Um, but it's a major breakthrough and milestone, if you will, for the Alliance and for the community at large. And a lot of the work we're going forward will be to try and fill these gaps and fund the research priorities to fill these gaps. So this was just a more detailed thing I'm not going to go into on the 5 working groups. I'm going to focus a little bit more on working group 3. As I said, despite the availability of more than 30 anti-seizure medications, there's high rates of refractory seizures, little known about optimal treatment. How do we improve therapeutics on a shorter time scale than realistic for novel therapeutic strategies, i.e. gene therapies? And the concern here is, does the registry provide enough information for us to improve a selection of ASMs, or what can we get from electronic medical records? And the registry's been ongoing, and there's been a project called Citizen that has conglomerated electronic medical records from any patient who agreed to participate. So the first thing I'm going to do is I'll tackle what we've learned from the Citizen data, the electronic medical record. We've been working with those data for over 2 years. We just had our paper accepted describing those results in epilepsy. We're waiting for the proofs. It should be available to everyone within the next couple of weeks. And I'm very excited about it in some ways. In some ways, it doesn't fill the gap, but step by step here. So, what you can get overall from these electronic medical records are, what's really nice that's different from the registry is we can get things like timelines, longitudinal profiles, because these records are based on office visits over the course of years. So while the data aren't fine-grained enough for some of the questions we have, they are longitudinal, long enough for helping us understand the progression of these diseases. So you can see, for example, seizure types over a period of 10 years. You can see growth for males and females, whether it's head circumference, weight, or height. And we actually find very big differences. There's declines in head circumference, for example, relative to neurotypical for boys and girls, but girls more so than boys. And so there's a lot of interesting pointers there that we need to understand, that are going to help us ask more questions and research. Comorbidities are, as we know, are vast, but we learned why do you go to the hospital? Why? These are all unplanned hospital visits, and this bar here is seizures, and these are non-seizures. And if you blow up the non-seizures, you can see it's infection, it's pulmonary and respiratory failure, pneumonia, infection, and other things, gastrointestinal. Anyway, we got an estimate of like why do you go to the hospital when you didn't plan to go, obviously, and that helps us understand the disease. But now I want to drill down on some very unique aspects of how we analyze the data. Again, with the question of how much information can we glean from these medical records with respect to improving treatments. And what we were able to do, and I will be able to provide these to the participants in the citizen study. We have individual profiles. So this is one individual, and you can see all their office visits over a period of 5 years down here in these green triangles. And then above that, you can see developmental milestones that are gained in green or lost in red. Above that, you have status epilepticus events in yellow. And then the seizure types that were reported in the office visits. And this patient had tonic, focal, and tonic-clonic seizures. And the red dots show you how severe that episode was based on what the patient told the doctor or the caregiver told the doctor. And above that, you have all the medications they were on for the full period of time. So here they started Keppra, they went to Oxcarb, they weaned Oxcarb, they added gabapentin, they had phenytoin, and then they switched over here in a major switchover event to valproate and rufinamide. And what we notice is there are breaks in the dot. The red dots separate out. So we call this a period of a reduced seizure burden or a gap. And so part of what we're trying to understand is what causes these gaps. Clearly, that's a pointer toward something getting better. And then we wanted to know how did it correlate with medications. We were also able to divide the seizure types over those 5 years and see how those changed over time. And then we have a heat map that says if it's red, it means a particular medication was added for a particular seizure type. So here's the tonic-clonic, the tonic, and the focal seizures. And you could see different medications did worse or better depending on seizure type. So levetiracetam made tonic seizures worse, more so than it made tonic-clonic or focal seizures worse. Valproate and rufinamide helped both tonic-clonic and focal seizures, focal and tonic-clonic and tonic seizures, not so much focal seizures. They have different patterns. So medications may help one seizure type but not another. And it's very important to see how this all plays out. This is just one individual. So we really wanted to be able to aggregate this data over all the individuals that participated in the study, and there were 80 of them. And I'm going to tell you what we learned about medications in a moment, but in the meantime, we learned about some other very interesting things. So here you can see, if you can see the whole thing, we looked at 3 years old, those with record, we looked at all of the individuals that had at least 10 years of data. And then we looked at those individuals. There was something like 40 individuals in the study that had 10 years of data. And we looked at them at 3, 5, 8, and 8 and 10 years. And we asked, we did this unsupervised clustering, which broke the individuals into different groups. So there was 2 subgroups here in year 3. Year 5, there were 5 subgroups. Actually, 4 are shown here. Year 8, it collapses back down to 2, and year 10, 2. And if you follow individuals through these grids, these sieves, if you will, you can see there are different subgroups within the population of individuals who had 10 years of data. And then when we looked and see what subgroup they were in, the DEEs were mainly in this blue ribbon. And so these were the more severe patients. There were some EE patients that started out looking like DE and ended up dividing with respect to DEE at some ages. And then there were some loss of function individuals and DE and EE individuals that were clustering together and separated out at different ages. And you notice at age 5, you have the most diversity. So the most diversity with respect to all these phenotypes. This is seizure types, developmental skills, EEG, all this stuff. What the conglomerate of features that patients have at these different ages. When you're 5, there's the most diversity. And then it kind of narrows back down when you get older. So kids continue to get more diverse in terms of the different array or suite of features they have. And then they kind of narrow back down, which is kind of interesting. And actually, one of the most exciting things for me is if you look at 4 domains of development: fine motor, gross motor, language, and academic skills, which we track also in the registry, but are also tracked in these EMR. You could see that for each fine motor, gross motor, each of the different subgroups tends to progress with age. This is 3, 5, 8, and 10. So for fine motor, loss of function generally achieve about 50% of the full boat of skills that are in the loss of in the fine motor category. DE patients do almost as well, followed by EE, and DE patients having the biggest struggle. And you look at the same thing with gross motor. Loss of function, kids with loss of function tend to do quite well. They almost achieve the full proportion of gross motor skills by the age of 10. DE close behind, and EE slightly behind them, and DEE, of course, struggling the most. Language skills, all the groups have some difficulty and really don't break more than 50% of what's expected at age 10, but getting there. The point is, on average, and the academic skills are quite interesting as well. Reading, recognizing letters, recognizing numbers, things like that, all the academic skills. And you can see this is averaging over within each domain. There's 4 to 5 to 6 skills, sub-skills that we used. And this is averaging for the whole developmental domain. So for the first time, we've been able to make a trajectory of what you might look like at 10. And we want to build a model to say, if we look at you at 3, where can we predict you will be at 10? That's one of our next goals, is to make models to predict developmental outcome, which I think is probably the most important concern of parents. Not so much seizures, or are they controlled? Yeah, the seizures are a nuisance and a pain and scary. But will we be able to develop and go to school and learn and maybe even begin to think about having a life where somebody can live semi-independently? So that's really one of the most exciting things I think we've gained. And it doesn't matter whether you're loss of function or gain of function. We've now got you on the same playing field, if you will, to see how you develop. So I think this is going to apply to everybody, whether you're mixed gain loss, whether you're fully gain or fully loss. I think this is going to be a very interesting way of going forward to try and refine this. This is just telling you the same thing, but it's breaking the skills down by individual subcategories. Running, for gross motor, you have running and walking and standing without support and sitting without support. And the earlier, more easily developed skills are in the top, the more difficult, challenging skills in the bottom. So we ordered the skills as well to figure out how far you made it towards the most advanced skill. So this is work in progress, and we're building models to do this better as we go. Now, getting back to which medications, I took a strategy of saying, what were the medication changes that it caused a reduced seizure burden? And I just want to point out here, this is an individual with epileptic encephalopathy. And you can see this is the one where we had the VPA, the valproate, and the rufinamide. I think for this individual, these 2 drugs made a difference. Not to make any suggestions to anyone else. This is one individual. So I'm going to show you the aggregated data in a moment. This is an individual with infantile spasms. Infantile spasms, we could not find or identify any period of time whereby there was a reduced burden of seizures. So there was nothing we could learn from what medications helped this patient. And it turns out that infantile spasms generally are a more severe DEE. They're associated with DEEs. They have a more severe outcome. And it's partly because they're hard to notice when you're young. And if you don't get on top of it when you're really young, right away, you could develop a whole bunch of seizure types that can be quite disruptive to development. And the average age and onset of these seizure types is like around 5 or 6 months. So we always, when we meet new patients, new families, we always we probe them to see, is there any chance that they're having infantile spasms, and we show videos to make sure you understand what they look like because they have their own treatment protocol independent of SCN8A, and they need to be stopped as soon as possible. Now, a good 20 to 25% of our population has infantile spasms. So why with one gene you have some people do not develop, some people do develop infantile spasms? What are the factors that lead to infantile spasms, which cut across all forms of epilepsy? It's not just SCN8As. And nobody understands why somebody's more prone to infantile spasms. We have found that there are certain parts of the gene with mutations that are localized that are associated statistically, highly statistically significantly with infantile spasms. So 850 is one of those. And then where Margot's mutation in the 217, the 210 to 220 region, if your amino acids in that range, I always suspect infantile spasms may be part of the picture. So here you could see this individual experienced infantile spasms here, and then the other seizure types that evolved thereafter. If you want, Madeline and I and some people at Boston Children, along with the Alliance, have set up this infantile spasms study, and we really need more people to participate. I think we've got 20 individuals who've signed up. You go to scn8.net, go to the registry, and it's module 17. It's called the Infantile Spasm Survey. It's pretty long, but it doesn't take more than 45 minutes, I think, to fill out. But we're really going to learn a lot about infantile spasms and how to treat those better, despite the standardized treatment. Like what kinds of things can we learn for SCN8A and going beyond that? Now, going back to the gap analysis, I told you I'd tell you about the aggregate. So of the individuals who we could identify gaps, and we only chose the longest gap, we said which medications were used in the gap. And it turns out for those on monotherapy, they all turned out to be sodium channel blockers, quite interestingly. Oxcarb was the most frequently used monotherapy, followed by lamotrigine, phenytoin, lacosamide, and carbamazepine. There were many individuals that were on dual therapy, and those are shown here in the purple, and polytherapy shown in the light blue. So the sodium channel blockers are over here, the GABAergic drugs here. This is for all the different mono, dual, and polytherapies. You can see Epidiolex was in the gap, but usually in the case of polytherapy or dual therapy, topiramate, clobazam. These are in drugs that seem to show up, clonazepam, in the gap. So they might have been doing something to help make that gap appear or lengthen the gap. Whereas the sodium channel blockers, the green shows you they were enough by themselves for those patients to create a period of reduced seizure freedom. So that's a clue. That's very interesting. But the problem we still face is that we don't have accurate seizure frequency. The average time between months of appointments for patients in the EMR was more than 2 months. So the doctor's going to say to you, how have you been for the last 2 months? And you're going to give an average, we've had roughly so many seizures per week. There's no good data. Even from the registry, we have an annual update. We're only getting an estimate annually. So I'm not going to I'm not going to talk about what Josh presented in Atlanta, but he's developed a model to tell us how to better select ASMs based on the registry data. And it's amazing that many of the same drugs that his model showed were best or worst were the same drugs that we found in the citizen data. But the nice thing for clinicians is clinicians have to choose from over 30 drugs. In the registry, it was primarily 15 that were mostly the first drugs. And so when you have 15 to choose from and you don't know what's best, you're going to have a messier outcome. And what Josh's model has done is narrowed it down to 6 that are probably the most reasonable first choices. Not necessarily which one or the order they're added or in what combination, but it should help with the next version of the consensus. If more doctors are choosing the same smaller subset of drugs, we should have a better idea of what's working. So I'm going to jump to what we're doing now to fill the final sort of gap in this regard of getting better granular data on seizure frequency in a prospective manner. You know, these studies that I've been telling you about are all retrospective. They're statistically not that as powerful as a prospective study. So we are now have a prospective study called using a daily seizure diary. It's a free app you can download, and I'm asking for people to we're still enrolling people into the study. It's a well-vetted, FDA-approved application for tracking seizures. There's a back-end data processing capability that we can tap into to do daily analysis. And we're then going to merge the daily seizure frequency and medication data with data from other sources for like EMR registry questionnaires, wearable devices. And we'll try and find out not only which medications seem to be working on this granular level going forward, but what correlates emerge. Do developmental skills improve when seizures get better, et cetera, et cetera. And so the app is called Helpilepsy. You can download it for Apple or Android. It's free. It supports both personal and research because you can take the data with you. We can provide you with a summary of your data to take to your doctor, what have you. And it gives us very granular data. So these are seizures and medications. You can see here's an individual that switched over to medications. It didn't seem to be a good switch because things got worse. Our prospective study, there's a setup period where you're trained to use it. And then the only eligibility are you've got an SCN8A variant and you have countable seizures every month. Just be able to count whatever seizure type it is. I don't care if you're gain of function, loss of function, mixed gain loss. I don't care. I just want you to be able to count seizures and have a medication that we can track at the same time. We'll track your developmental skills. We'll track the child's mood and sleep. We'll track your quality of life, et cetera, to see how things change over time going forward. It's very consistent with FDA guidance, which is switching more to not just listening to the clinician, but listening to the caregiver. Your assessments of outcome matter as much as seizure reduction by itself, which is all the clinician generally measures. We're going to have a composite outcome measure that we think will be very useful. This is, again, the app. We can tell you more about how to get it and how to use it. Right now, we have these people are enrolled in the study. This is where their variants are. We have a nice assortment of people with different variants, some in the infantile spasm range. We need a few more people. We have 2 more people to join that have a variant in this region and have infantile spasms. That's really good. We have a bunch with the 1872 cluster, and we have 2 patients with a splice site variant. We know very little about how splice site variants work and what they cause. It's much more complex than standard missense variants, whether they're gain or loss. This is going to give us a very mixed gain loss picture. We have 2 with the same variant, which is fantastic. We'd like more to enroll in the study to increase our numbers. And we'll give back to you every month an infographic that tells you how you're doing, how your seizure-free streak is going. We'll tell you what other patients are in the group that have the same variant. We formed a WhatsApp group so we can talk to each other. And it's all very exciting. And I'm going to end there because it's coming up on 55 minutes. I want to thank all the folks here doing the hard work, the data analysts, Elise, the clinical coordinator, of course, the Alliance, and how much they're contributing. Principal investigators in math, statistics, John Schreiber as a clinician, we often resort to for clinical questions, support from Neurocrine Praxis and the Shea Hammer Research Foundation, and where I work at the University of Arizona. And of course, the families themselves. We can't do any of this without you. We love you. We are you. And fortunately, we have a very nice group of members of the community that are now a growing number that are scientists and have other skills that can help us understand this. So we welcome your support and your help at any level that you can provide it. So I'm going to stop there and open it up for discussion. I'm going to land on the last slide in case you want to try getting one of these apps or getting the Helpilepsy app and joining us. I'll leave that up for a minute. You could use the QR code to go directly to get it. And once you get it, just email us at info@scn8a.net to let us know, and we'll get you started.